Hello, everyone. We are lucky to have with us a true scientist, Professor Dr. Mushtaq Hussain. He is currently the principal of Dow College of Biotechnology at Dow University of Health Sciences, Karachi. Dr. Mushtaq obtained his PhD in genetics, genomics, and systems medicine from the University of Glasgow, UK. I worked with him, or should I say, under his mentorship between 2014 to 2016 on a project related to evolution. That project was funded by European Society for Evolutionary Biology. I learned a lot from him, including but not limited to what research really is and how to think critically. His scientific discoveries and achievements have earned him many national and international accolades. He has been working on common fruit fly Drosophila. Please welcome Dr. Mushtaq with the question, why Drosophila, or rather, what is Drosophila? What intrigued him to explore this creature? Welcome, Dr. Mushtaq.
Thank you, Jamshed, for such a nice introduction, and thanks for having me. So, why Drosophila? So let's say it dated back when I was doing my BSc in 1998. So in the we have three subjects in the BSc. There is a chemistry, zoology, and microbiology. In the zoology, there is an experiment regarding how to rear Drosophila at home or in the laboratory condition. And what they used to do, that we used to make a pulp of the or plump of the banana and leave it at any place, and we hope that the Drosophila will lay the egg over there and it will then hatch into the larvae, and the larvae is eventually going to form the fly, pupa and the flies. So that was a little background that I already have, that how to rear Drosophila from the environment. But since then I stopped, never thought about the Drosophila, never think about the Drosophila. And then I've done quite many different kind of the jobs, different kind of the research projects. My PhD project was completely different. That was about the Epstein-Barr virus. That has nothing to do with the Drosophila. Neither follow-up research experience of mine have any component of Drosophila in it. But there is one thing which was very common since I started my PhD. And what was that? Well, yes, I was doing my PhD at the University of Glasgow. There are quite much of the facilities being available over there. If I have to ask for a consumable, it will be ready for me on the very next day or next to next day, right on my bench. That I realized from my past experience while working in Pakistan, either at the AKU or at the Karachi University or at ICCBS, that at times the consumables came very late. It takes about a month, 2 or maybe 3 to be delivered in Pakistan. Number one. Number two, they are heavily expensive. They are extensively expensive. So when I was doing my PhD at the University of Glasgow, that thought was actually in my mind, that when I'll be back in Pakistan, whether I'll be able to secure the funding or not. And if I'll be able to secure the funding, how much funding I'll be able to secure. Number two. And if I'll be able to secure that much amount of the funding, how much sustainability I can get out of this funding. Like this may be a one-time installment, or it may be present for 3 years, and what happens afterwards. So I need to learn the skills which, despite of lack of financial resources, or even the physical resources that might the lab or may be present or may not be present, in that case, I can continue with my science. And for that purpose, I started working on the bioinformatics part. That's the computational biology. And about what I have found, there's a big advantage in it. All you require is a good internet connection, a good laptop, and few working neurons. And if all these three things are present, you may be your science is not going to die just because you do not have any lab, or you do not have financial resources. So I started working on computational biology. Part of my PhD is actually based on computational biology, precisely because of that reason, that in Pakistan, whatever happens with the financial resources, whatever happens with the physical resources, my science is not going to die if I learn those skills. Well, the idea works when I'll be back in Pakistan in 2014. And till 2018, 17, 18, I was precisely working in a hybrid kind of a research or standalone bioinformatics. So there is a part experimental, part bioinformatics, or there is entirely bioinformatics kind of a work. But what I realized, or I actually learned over the period of time, there are few major flanks in any scientific investigation. One of the flank which we call in vitro studies, which we do by lysing the cell, extracting the molecules, either protein, carbohydrate, nucleic acid, lipids, from the cell, and we conduct those experiments on the laboratory bench. That's in vitro flank. In silico, that is a bioinformatics, computational work. Then we have ex vivo, we have cell culture system-based studies. Then we have in situ, that is the histology-based studies, where the tissues are dead, but their structural architecture is already there. So so these are the four flanks, and one of the major flank, the bigger or the gold standard flank, which we call, is actually in vivo. That is doing experiments in live animals. Would you please excuse me for a while? Here's my cat, actually. That's Leonardo.
Welcome.
Sit down, Leonardo. He wants to be an audience. Okay. So these are the 5 major flanks of any scientific investigation. And over the period of time, from 2014 to 2019 somewhere, I've already established in vitro system, a preliminary primary kind of an in vitro system where we can extract DNA, RNA, proteins, and we can run protein gel, DNA gel, we can conduct thermal PCR amplification and things like that. So basic fundamental ingredients of in vitro research are available. And in situ, we are continuously doing it by taking histological section of different kind of the human cancers and monitoring the expression of different kind of the protein in those cancers. The third thing is in silico, that is already in my laptop, in several of the computers which I made available in my laboratory. And ex vivo system, we have already started on working on the cell culture of diffuse large B-cell lymphoma. So I always wonder what is being lacking, and the lacking part is actually in vivo system. And you may call me a bit possessive in nature. I want all of those things under a single roof. So I made in vitro system, in situ system, ex vivo, in silico under a single roof. But when it comes to in vivo system, that's a bit of a problem. Because if you want to work on the live animals, and consider you want to work on a live animal like 100 mice, there is an experiment you design on 100 mice. How could you put it in front of you all the time? So that's a difficult bit. And next, they are expensive. They are difficult to be maintained. So there comes my zoology experience of 1998, where I have learned how to grow Drosophila in simple laboratory, or in simple at home even. So on 5th February 2019, that was a Kashmir day. It was a holiday, and that was the last holiday possibly that I was at my home. So I said, oh, why don't we start working on Drosophila? So there are two brilliant BS students which were working under my supervision at that point of time, and we were thinking about doing some work on bacteriophages. And on 5th February, I was at home and I have nothing to do. At that point of time, I don't have cats as well. So I have pretty much nothing to do. So I put two or three jars of banana at different places in my house. And I leave them. At about 25th of the February, I noticed there are pupas there. So I say, all right, whatever so Drosophila might have come to those jars, lays the egg on the banana, the larvae came, the larvae made the pupa, and now I got the first founder culture. I brought those jars in the laboratory. I allowed them to propagate for us for about 2 to 3 months. And now I got a seriously huge population of the Drosophila right in the laboratory. So now all 5 flags are present. That's an in vivo system now being established in the Drosophila. So that's the matter of convenience. But while doing so, over the period of time, I have read few books. And one of those books is actually this Gilbert Developmental Biology. And that I have read during my PhD, this book. And when I read that book, I realized that Drosophila is one of the very simplest models to study developmental biology. That I am aware of, that quite much of the genetics has been done on the Drosophila. But then the modern day genomics and modern day molecular biology and modern day developmental biology is also being done in the Drosophila. Even so much so that most of the genes which are being embryologically imported for the development of living organisms, particularly eukaryotes or multicellular organisms, are fundamentally being discovered in Drosophila. And they've been named in the Drosophila, and later you identify the homologue of those genes in the human, and you name those genes exactly the same, but the human version. Roughly speaking, what we call orthologs. So that's the scientific basis while those Drosophila was propagating inside in my laboratory. And then I realized, then we read a lot about Drosophila, and I got lucky actually. At that point of time, I got 2 very brilliant, very hardworking students with me. If they won't be there, I don't think so I'll be able to start Drosophila. I would be able to sustain even Drosophila, or I would be more interested in working on the Drosophila. Fortunately, I got 2 very brilliant BS thesis students that is Anusha Amanullah and Sharon D'Souza. They both have worked very hard, and it actually is, rather than me driving them, they are driving me more and more into the Drosophila. And a time comes when I wrap up all the other researches of mine. I have nearly wrapped up all other researches. I have worked on the human cancers. I have worked on the various kinds of the dental diseases. I've worked on aggression. I've worked on, let's say, different kind of the phylogenomics kind of a stuff. I have left all those things. And that 1 year experience of mine with Anusha and Sharon and with those little fruit flies intrigued me so much that I have dedicated the entire lab of mine to the fly work. So if Jamshed allow me to rotate my camera, then I can show you some of the basic things. Am I?
Yeah, we'd love to. We'd love to.
So that was the first incubator. The one for this incubator was the first one we bought. And that incubator was not that glamorous at that point of time. It was a very dull incubator. It has only 1 bulb in it. It's just to have a light. And since we were ill-experienced at that point of time, so we sustain it like that. But then over the period of time, over the experience in the last 3 years or 4, we have learned a lot that how to actually work with the flies, and we custom modify every single incubator. Then we subsequently buy this one, and then we buy this one. And these are the normal incubator which we custom modify for our work. And then we got the big one there. So
Excellent.
most of the experiments are being present there or there. this incubator. While these two incubators act as a factory for propagating Drosophila. So as I was saying, that mice could be a good in vivo system, but for that, I require a separate animal house. And once I have a separate animal house, then it's not under a single roof concept. And you may call me a bit possessive. I want all of those things to be right in front of my eyes. So that's why. So there are quite many things, factors that allow me to work on the Drosophila, that push me towards the Drosophila. The students, finances, all 5 flanks of the investigation, and convenience. So, but the most important factor is that, as I have already said, that I got a very nice undergrad at that point of time, then undergrad students, Anusha and Sharon. They worked really very hard, so that allowed me to continue working on the Drosophila, so much so that I completely obliterated or nearly wrapped up all the other kind of these studies or research I was involved in. All microbiology is gone, all other stuffs are gone, cancers, etc., all are gone. The only thing which is now left is Drosophila. So, and Anusha seems still be here, and that's where the Anusha is. If you can see, I'm not the alone in the lab. Anusha, that's the girl. Just wave. This is the girl. That's Anusha.
Excellent.
So she's nearly ending her MPhil now. Very fantastic work. She's working, very fantastic project, very nice findings. So, yeah, that pushed me to keep continuing with the Drosophila. And now we got a very fine lab, and I'm slightly pride and bit arrogant in saying this, that this is the second lab in Pakistan that is being exclusively designed for Drosophila work. The one is at the LUMS, and second at Dow University of Health Sciences.
Brilliant, Dr. Mushtaq. It's so amazing to learn the reasons, for example, logistical reasons, and then you have the team. You are lucky enough to have this team, and you are, of course, hardworking, and the scientific reasons for studying Drosophila. So you studied on variety of organisms and variety of fields. And the underlying theme that joined all of them together is the theory of evolution. It is the evolutionary perspective, evolutionary knowledge that allowed you to join all these fields and then focus strictly on Drosophila. So regarding that, we have a book club member, Rashid. He is a third-year student at Salim Habib University. He would like to ask you a question regarding evolution. Ji Rashid, you can please ask the question directly.
Assalamu alaikum, sir. I have one question regarding the theory of evolution, that many people still not believe on the theory of evolution right now. So which steps should be taken according to you that they should consider this theory of evolution is right related to theory of evolution of mankind?
Well, I can understand where this question is stemming from. And I was in that frame of mind as well. But now, over the period of time, as Darwin has actually said, things evolve. I also evolve. Possibly Jamshed might have evolved as well. That we do not here in the business of, you can say, preaching evolution. All we can present the facts, and those facts, if they are aligned with it, they are okay with those facts, and if they can value reasons, there is no doubt in my mind that if somebody is valuing reasons, they can simply deny evolution is not happening. They can't. And if somebody is still, after looking at all the evidences and still denying it, rest assured, in about 95–99% of cases, they have made up the mind. They are not going to believe you. There is no reason of schooling them. Let the nature tell them. let the ignition comes from inside rather than you forcing them. The more you will force them, the more encouraged or more stubborn they would be on their beliefs. And given the society which we are living with, is आप भी जितने अच्छे मुसलमान हैं उतने ही अच्छे मुसलमान जमशेद भी हैं, उतना ही मैं भी हूँ और उतना ही कोई और भी है. लेकिन यहाँ पे जब हम आर्ग्यूमेंट्स बेटा हारने लगते हैं ना, तो फिर we have other repercussions of it. So it's better not to get into such things. Does it make a difference to you that if somebody is going to believe evolution or not going to believe evolution? I was very enthusiastic at one point of time about teaching evolution and making people convinced that it's a really interesting field. You need to see into it. You need to cherish about the facts which of the evolution. And if you see all of those things, you find all of those being related to each other. And later I realized my approach was wrong. That the students or the individuals around you may have their personal truth. And I have lot much other things about science to tell them. And if I strictly talking down about, oh, this is evolution, this is interesting. Okay, that's interesting to me. That is very interesting. I can understand that. It's a bedrock of the entire life sciences. That's pretty much true. We do experiments either on Drosophila, either on mice, or either on monkeys. Why? We test those experiments, we test any drug, or why we test the effect of any chemical on any animals. For what reason? It's because we share some common genes. And why do we share our common genes? Possibly because of the common ancestry. And that story is being written in our DNA. And I'm absolutely sure that we can't deny what DNA is telling us must be very accurate, because if we are going to deny that, then entire of the forensic sciences or entire of the forensic biology is out of the window then. So if your DNA is telling that these are, you share your common ancestry with your parents and then your grandparents and then your great-grandparents, that's been written in your DNA. And the same approach, the same DNA is telling you that we do share the common ancestry with all other life forms, including even banana. Then it's the same approach. Denying it would be denying entire of the forensic science. But then I realized over the period of time, if I dictate too much about the evolution and I said that, oh, you need to believe on it, then they might not be interested in listening me in the other interesting facts of the science. So once you can say hook, line, and sinker, you can expose them to the different branches of the science, and then they come to you. They automatically open up the books and they will see, oh, they are all related, and gradually the plant will grow, and they will be convinced about the evolution. And it's not about being convinced or not convinced. It doesn't make a difference. Whether you believe it or not, that the earth rotates around the sun, doesn't make a difference. It only makes a difference to you. It doesn't make a difference to the rest of the world, or those who are designing rockets, or those who are developing drugs. You believe it or not, or you know it or not, or you are convinced with it or not, evolution is not going to change anyway. So yeah, everybody be happy, believe whatever you like. Let's see how far you can go from it.
Okay, fantastic. We have Aida. She is a student of Dow College of Pharmacy. She has a question. So Aida, please go ahead.
I have lots of questions, but पहले मैं सिर्फ अभी एक ही question करूँगी. Well, your study on Drosophila, like it's like fantabulous, now, and I had many questions about that, but you answered most of them. But I'm curious, like the amount of study you have done on Drosophila, are there any cons to this study? Like Drosophila model, is there any con to it? or is there any disadvantage that anyone can face if he or she goes into that area of research now? I'm talking of from the perspective
I
of
think so.
clinical research discovery, drug discovery. So that's my question.
Yeah. So the first, and take it as an advantage or disadvantage, different which reference, which frame of reference you are speaking to. As I said that I put the first banana jar at my home on a holiday. That was 5th of the February 2019. And little at that point of time, I know that this was possibly the last holiday I am having at my home. So that's the first disadvantage. If you started working on the flies, make sure you have a very, very dedicated team, that somebody is over there on holidays to maintain the culture. Fortunately, I did have, but that's the first disadvantage you may get, or maybe an advantage if you are being an introvert and you don't like people much, like me. So neither they do like me. So we are being under a common terms. So we have, me and other people have a mutual affections. Neither of us like each other. So that's the first disadvantage. Now the other disadvantage. Well, with any animal model, there are disadvantages associated. Absolute disadvantages associated. Say, for example, consider the best animal model you can pick is a chimpanzee, isn't it? Because that's the most closest to the humans. If you want to test any drug, in fact, all the SARS-CoV-2 vaccines have been tested on chimpanzees. 98% of the genome is being similar. So why not chimpanzee then? But chimpanzees are present on a very few places around the world. Their population is not that much, and plus their generation time is in terms of years. And they are going to lay 2 or 3 babies, or maybe 1 baby at a time. And maintaining them is one hell of a job. The area you may be requiring it. But you may say that some of the humans may behave like a chimpanzee, and they could be used for that purpose. But quite frankly speaking, there are ethical concerns for that. And even if you are using chimpanzees, there are significant ethical concerns for that. And those concerns are better relaxed when you go to the rabbits or mice, and then better relaxed then you go to the, let's say, chicken, or then you go to the frogs and fishes. But Drosophila being an, so that's, you got an advantage over there because of the more of the similarity with the humans, but at the same time, you got a disadvantage with reference to the numbers of the replicates you may be able to run, just sheerly because those animals are not propagating well, not propagating as fast or as in as many numbers as in the case of the Drosophila. So you got an advantage on one side, but then you got a disadvantage on the other side. Now those advantages could be compromised when you are going to use Drosophila, because Drosophila share only 60% of the genomic similarity. But 60% is a whole lot more than 12% or 10%. And 75% of the human disease genes have a 77% rather. 77% of the human disease genes have a direct orthologs present in Drosophila. So that's the degree of the resemblance we are talking about. One of the classical way of showing that how closely related to the Drosophila is that we got organ after organ, tissue after tissue, and genes after genes, which are analogous, homologous, and orthologous to what being present in humans, and that's been present in the Drosophila. So let's say, put it in this way. So if you like to see, this is, may or may not you be able to see. This is the glass slide. And you can see there are few blurry pieces in it. Can you see that?
Yes, yes, they are visible.
Okay. They are actually the wings of Drosophila. These are what? Wings of Drosophila. Now, those wings of the Drosophila has small bristles. Say, for example, if you're traveling in a plane and you see only one tree and another tree and another tree and another tree with a distance of about 100 meters each, you will find, if you are going to observe from the top down, what you are supposed to see, one green dot, another green dot, another green dot, another green dot. That's how it will appear. So the bristles are being far apart from each other under the microscope. They look far apart. One here, one here, one here, one here. But if, say, for example, many of those trees are being clustered together, then you will find a whole blob of green. So when the Drosophila are exposed to any carcinogenic agent, what happens? From a single point where there are supposed to be one or two bristles, there are multiple bristles coming out. And that makes, instead of a point, a dot. Rather than a dot, it makes an entire big spot, the black spot. Now, it's a very simple observation, but think about the possibilities there. So the Drosophila could be the biosensor of a presence of a carcinogenic compound and anything, anywhere. It's a very simplest assay. Now, you can run a whole lot of a big, expensive chemical assay and a cell culture system. And Jamshed must know, Jamshed is very well aware of how expensive the cell culture system is. Okay. And how much amount of the facility you may be requiring for that purpose. And the skill set you may be requiring for that purpose as well. Now, you can detect a carcinogenic agent in a cell culture system. Okay. But then it's an ex vivo system where the cells are being completely exposed. Obviously, they are going to be behave normally in the presence of the toxic compound, because all of their covering, their skins are gone. They are out naked. There is no sheet of protection. Then you will say, all right, treat them with the mice, but then there are ethical concerns. And then how many mice then? 100, 200, 300, you see, as many as you increase. You got another problem. The problem is how to maintain those 200 mice. And say, for example, you are a single PhD student or an MPhil student. And what's your name, beta? Aida. Aida, okay. So either, let's say you are a postgraduate student and you're working on 200 mice, 300 mice. Trust me, your mother is going to kill you. And if you are being married, your husband is going to kill you. 300 mice is not manageable in a single day. You can't just see those mice, 300 of them in a single day, if you want to see them properly. Either they are getting cancer. Think about it. Each mice you are picking from a tail, completely scanning each part of it. There is a small tumor, big tumor, anything like that. All right. And you can't just, and say, for example, I start putting those mice in formalin, then on a single day, all 300 are gone. And then you have to rerun the experiment for the 300. And then you need space to store those mice. Now, compared to that, those 300 flies could easily be maintained, could very easily be maintained in, let's say, if you can see, now, if you see around here, this is a jar, and this is a rack. You may see the wires there, and you may see few pupas. Can you see?
Yes, we can see. We can see. Yes, visible. It's visible.
Yeah. So in each of those vials, there are about 40 flies which will come in about a day or two time. Okay. So think about it. It has 50 vials, 40 in each, 40 in each. Think about it. So 50 multiplied by 40 is 2,000 flies in that frame. And I could accommodate about we have calculated it that we could run about 6,000 vials in that incubator. 6,000 vials in that incubator. Each have 20 flies. Each have 40 flies. So imagine. So that many number of flies, that many number of replicates you can run. And as you all are well aware than me possibly, that more the replicates in your any scientific investigation, more confident, more robust your analysis would be considered. So if you conduct an experiment on 5 mice, compared to that, you conduct an experiment on 50 mice. The study, the data, no doubt the 5 mice result is very much aligned to what your hopes are. That, oh, the toxic effects are there, but when you run that experiment in 50 mice, you did not find that much kind of good results. But those 50 mice are the better telling the actual story rather than the 5 mice. So in that case, we could run an experiment on about, let's say, 6,000, 8,000 flies. And beside, it is very easy to trace where it is leading to, because the generation time of fly is mere 10 to 12 days. Into 10 to 12 days, you got the first progeny from the parents. In the case of mice, which is the most commonest of the laboratory model animal, which we generally know. So you hope, if you are running for a genetics experiment, or if you want, since I worked on transgenic mice during my PhD. So when I was so it's not like that I started working in the Drosophila, it's the first animal I have been exposed to. No, my PhD is on transgenic mice, it's partly on transgenic mice. So we made some transgenic mice out of it. So then we hope that this particular mice mated with this particular female, and hopefully they would give good 8 or 9 pups which are healthy, and hopefully out of 8 or 9 pups, 1 or 2 might have my transgenes, and hopefully they could be a pair, so I could then mate them, and then hopefully there would be baby. So in this way, it's a long exercise to have a line of a transgenic mice. So having a line of a transgenic mice, it might take 2 to 3 years, and then the validation may require further 1 to 2 years. In the case of flies, I remember last year, yes, I guess it was last year. Last year in September, Anusha's grandmother was in a hospital, and she was here, and she observed there was a fly without a head, and it's moving. So Anusha, as usual, she has a very nice observational power. So she collected that fly, that how on the earth a headless fly is rooming in a founder jar. So she collected it, and that turns out to be a male. And it was not headless. The eyes of that fly is white. So the Drosophila has a red eyes. So it's a large red eyes. So their head has been recognized precisely because of the eye. The red dots, the 2 red dots. Okay, that's the part which is the head. If those red dots are white, then it appears like it's a headless fly. So then we observe it under the stereo microscope, it turns out to be red. And now, do you remember, at that point of time, her grandmother was in a very critical condition in a hospital. She collected that fly, she bred them with the other, specific kind of the crosses have been made. And now we got a whole line of Drosophila flies, which doesn't have normal red eyes, which have white eyes. They're all white in eye color. And these lines are very good to be the white eye flies. There is another line which have been developed very early on in the Drosophila research around back in, I guess, Aisha, ये अपना W118 30s में बनी 1930s somewhere. W118 has been there's a white eye fly that has been made in 1930s by T.H. Morgan and his students. That our white eye is slightly different from that white eye, but that the white eye which the T.H. Morgan has developed, now it is being known one of the best model, even much better than mice, for studying neurodegeneration. So may I have answered part of your question?
Amazing, amazing. It's so brilliant. We have received a question, and part of it was Rashid's own question. The question is, can you suggest or recommend some books to study evolution of mankind? One of the books that you mentioned was Gilbert's Developmental Biology. So what are some of the books that you would recommend someone if they are interested in evolution of mankind?
Human evolution.
Yeah, specifically human evolution. They want to know about themselves.
What are the evidences which are being raised? So scientifically speaking, as far as peculiarly, I did not. You may go to the Alice Robert Evolution, the Human Story. That is one book which I would like to suggest to you. If you got a broad picture that what scientific evidences we have as far as the human evolution is concerned. But let me tell you, that's a more of a Alice Robert Human Evolution Story. Yes, that could be the one thing. But it will tell you the story most in the anatomical perspective or paleontological perspective. There is a part genetics in it, part DNA in it, but mostly it depicts how the anatomy has evolved, or how your bone architecture or your posture or the social network of humans have been anthropological. Anthropology has been evolved over the period of time. But if you want to go for the DNA thing or the genetics things, pick any good book of the human genetics. Pick just any book of the genetics. And you will know it by yourself. Or just pick up any gene sequence or protein sequence of any animal, blast it, search it within the human genome. There are databases after databases being established for that. Or pick any human gene and blast it in any animals and see what happens. Don't believe on me, don't believe on any other book, see it yourself. Just pick any gene of human, blast it in Drosophila, and pick any gene of Drosophila, blast it in human. See what happens. See what happens. And if you find similar sequence, now obviously, Rashid, if I pick you and your parents and Ida's parents, and I pick, let's say this, another girl or guy, Ghasif and Sundas. So say, for example, I bring Sundas and I ask them that, can you identify the parents of Ida? Or can you identify the parents of Rashid? She may be able to do so. And why she may be able to do so? Precisely because of the reason that she would look for the similarities in your morphological attributes with your parents, and similarly Rashid morphological traits with his parents. And based on those similarities, she may be able to identify the parents of yours and the parents of Rashid, or the common ancestor, your common ancestor and Rashid's common ancestor. Okay. Now, So similarities denotes common ancestry. If we are agreed with that, then those similarities should be present. Since we say all of our traits are coming from the DNA, so yeah, that's the same story which your DNA is going to tell. Even I do not have your photographs, all I have your blood samples. 2 of your parents and yours, 2 of the Rashid's parents and Rashid's blood. So 6 blood samples, and out of those 6 blood samples, I take out the DNA, and the DNA is going to tell the same story. Because we say all traits are coming from the DNA. So it means that we do not need fossils anymore, or bones, or any, well, paleontology has its own significance. Absolutely, it's no need and no reason I have to deny the significance of paleontology. But then, having said that, it's simply the DNA is going to tell you a whole lot of evolutionary biology. So those resemblance within the DNA as your traits are also going to show the common ancestry. So as far as human evolution is concerned, there's one book which you can see, and if you want to go for the genetics or genomics side, then quite clearly go for any book of the genetics. Say, for example, there's a very fine book of Human Molecular Genetics. If you pick that, that's a brilliant book to read about the genomic similarity between the humans and other animals. An entire of your forensic sciences actually has been based on it. Any book of the genetics, for that matter, like this book, which is a common textbook in many of the leading universities around the world for the genetics. This one, in most of the, let's say, Australian part, this is the book which they generally refer genetic analysis. And similarly, that's the book which I was referring to, if you want to go for the humans one. But that will give you more of an anatomical perspective, let me tell you, because this is Dr. Alice Robert. And Dr. Alice Robert per se, she's a professor now, professor of public understanding of science at the University of Birmingham. So she's a medical doctor by training. A medical doctor turns into an evolutionary biologist or anthropologist. So that's unique.
Excellent.
Thank you, sir.
I have one question. You described that if you don't believe in evolution and if you don't understand and you say that, okay, it's just a myth and you don't believe it, fine. But the developed world, let's call it developed world, they have a professorship of public understanding of science. So they think it is very crucial for a common man to understand what science is all about. So I would like your comments on that, why do they have a special professorship just for this purpose, right? So your comments on why public understanding of science is important. One way of thinking about it, as you mentioned, is that, okay, believe whatever you want to, fine. But another attitude is, yes, I am concerned that people do understand it. Just like one of the projects that you helped me in understanding. That project was on evolution, and we went to various universities and talked about, understanding of science. So you also believe in public understanding of science. Why do you think it is important? And it is important to, dedicate a lot of resources for it. So some comments on that.
Okay, absolutely. The public understanding of science is an extremely, possibly the foremost important part for we being a progressive nation. There is no doubt in my mind, absolutely no doubt in my mind. But then I have to loan some of the wordings of Neil deGrasse Tyson. So what he said, and he put it very nicely. Look, there are three kinds of the truth. One is a personal truth. One is a personal truth. And the other truth is a social truth or a political truth. And the third is an objective truth. And impact generally comes when all of those three truths are living, despite being disagreeing with each other, living amongst, living with harmony, or they are being perfectly aligned. Say, for example, many of the European countries, if you like, then you will find their objective truth, their scientific truth, and their personal truth. They are all aligned. I do not want to go into the specifics of it for very obvious reason. But say, for example, my personal truth is that I believe on Almighty. That's fine. It's my personal truth. And that Almighty, for us, it may be Allah Ta'ala. And let's say for somebody else, it may be Jesus, or for somebody else, it may be some other God, and some other God, and some other God. Okay, fine. You all believe on it. And somebody don't believe on anything. That's okay. That's their lives. For me, that's their personal truth. And I do not feel myself being capable enough to challenge their personal truth until and unless it is hurting me, or it is hurting my child, or hurting other way around, my lab or my scientific investigation, or whatever you like. So at that moment of time, the personal truth doesn't make a difference. Social truth is one thing. Say, for example, if you go to the States, you will find, despite of the scientific, overwhelming scientific evidence, about 50 to 60% of their population and about 80% of their states do not agree that the global warming is because of a human intervention in the ecosystem. They believe it's because of some divine plan. It very well may be, but then the human intervention is there. And it could be, and once you agree that there is a human component into the global warming or whatever the climatic perturbation we are getting, if you agree on that, only in that condition you try to think to rectify it. So now your social and political truth are actually hurting us of resolving problems. And that's where the problem. If your personal truth are generally not harmful, but if you make your personal truth as being a political truth, you need to believe on that. And similarly, if somebody wants to believe on evolutionary biology, that's fine. I agree with the concept of the evolutionary biology as far as the science is concerned. If you ask me personally, well, that's my personal thing. What is my personal belief is, it has nothing to do with my scientific investigation. May I believe personally evolution, or may I not believe personally evolution? But if scientifically, when I'm holding my test tube, when I'm running my DNA, when I'm looking at the Drosophila, or when I am observing flies under the microscope, or when I'm teaching my students, I should be honest enough. And that's what the teaching of our religion, if you like. I should be honest enough to what's being written over there, and whatever the evidences they are presenting there should be imparted to these students. And if there is a counter evidence, I would love to give it to them. But at this moment of time, we do not have a credible counter evidence for that, credible scientific evidence. Correct. And then the third part, it comes from an objective truth. As I said, the objective, this is what not my statements are. These are what the thoughts of Neil deGrasse Tyson. So He said that the objective truth are those who are being investigated by continuous experimentation, rigorous checking, and then rechecking, and then rechecking, and that comes out as an objective truth, which will say this is right, this is wrong. So now all objective truths are leading towards the global warming. One of the major causes of the global warming is actually the over industrialization, agriculture, human intervention in summary. So once you know those problems by objective truth and make an objective truth being your political and possibly the personal truth, then you start resolving the problem. So any position of public understanding of science is primarily driven to make those controversial objective truth who are not generally being aligned with their personal or political truth or social truth being aligned to it, and so that they could look for the problems. So yes, if you ask me, public understanding of science is heavily important. I would say it's the foremost requirement. It's a foremost requirement. If we want our next generation to have some al-Biruni in it, or some al-Khwarizmi in it, or some Abdul Salam in it, if we want a next Isaac Newton to be born in the Muslim world, yes, the public understanding of science is very important. And I would say how distasteful it may look to you with your belief system or whatever the philosophical connotation you might holding to. हमें ये कड़वे घूंट कुछ पीने पड़ेंगे if we want. ये सिर्फ कह देने से नहीं होगा कि हम अल-ख्वारिज़मी बन जाएंगे, हमारे ये थे, हमारे वो थे, होंगे, ठीक है, but where are you right now? And if you want your next generation to have some of it, then quite clearly कुछ कड़वे घूंट आपको भी पीने पड़ेंगे. ये मेरा ओपिनियन हो सकता है, मैं गलत भी हो सकता हूँ. I mean, you may completely disagree with me.
Yes, you're right. It is your beliefs and thoughts that ultimately lead to action. And if that action is destroying the environment, including other people in it, then you have to worry about other people's beliefs and their thoughts. So that is why public understanding of science is important. If public don't understand science, then they will believe in all sorts of wacky things, and those sorts of wacky things will disturb even the existence of human life on Earth. So, Rashid, you wanted to ask a question?
Yes, I have one question. Sir, you are working on Drosophila and आपने यानी कि different crosses भी कराते हैं, तो इस केस में cousin crosses अगर आप कराते हैं तो उस केस में आपने कौन सी ऐसी mutations या abnormalities observe की हैं first cousin, second cousin में, और then क्या वो abnormalities जो Drosophila में आपने observe की हैं are they linked with humans?
Okay. So that experiment is nearly culminating right now, and in which we have actually run, if you like to say, 6 different kinds of crosses. One is an outbreed cross, and the other one, the other kind of the crosses are 5 different kind of relationships, like siblings, cousins, first cousin out, and then sibling out, and then again sibling. So it's different kind of the related cross, and we are running it for 5 generations, and obviously we are finding quite interesting results out of it, and in which we are observing there are errors. They are not only errors with reference to the morphology, but we are also observing that there are errors with reference to their behavior. We are also observing the errors with reference to their learning capabilities, which are being damaging. So there are some assay systems. Let me give you one example. So there is a girl over here, Aisha. She actually ran Aisha. That's Aisha there. That's Aisha. So what she has actually conducted some experiments on learning assays of flies who are being subjected to different kind of the crosses. That includes cousin crosses. And this is my cat, which is actually. So what she, then there are 5 different kind of the relationship process. One, cousin, siblings, and so on and so forth. And there is one type of a cross, which is an outbreed, where the flies are not completely unrelated to each other. What Aisha has conducted an experiment, which is called as Aisha along with Anusha, conducted that experiment is called photo-aversive assay. And what it shows the learning behavior of the flies. And what we observe actually that the learning behavior has been affected. To which intensity, that you will wait for the paper. And how we identified it, that their learning behavior is affected, that you will understand. So learning is one of the important things as far as the humans are concerned, isn't it? Your child, the first thing you want in your child, that he or she would learn how to walk, how to talk, how to recognize his name or her name, isn't it? This is one of the requirement, the fundamental requirement or fundamental wish of the parents to their offspring, is it? So if the learning has been affected, and that's a very complicated neurobiology involved in it, which Jamshed and Humaira might have worked on the neuroscience, so they are well versed compared to me. So there's a very complicated neurobiology has been involved in it. And if the learning has been affected, imagine what are the other possibilities. So it's all there. There are quite many errors. So we got a fly which have no brain in it. So there is entire head, eyes are there, cranium doesn't have a brain. So there's a human condition which we call encephaly. So say for example, Ida may have heard about this condition since they go through the anatomy. There's a book, Langman or KLM or any other book of the embryology, if you read, you will find there is a condition which is called as encephaly. So you got to fly, all things are normal, but the brain is not there. Brain simply failed to develop. So these kind of the errors are there. Similarly, the wings, a part of the wings are orthologous to our limbs. Part of the abdomen is orthologous to our abdomen, and so on and so forth. So if you want to find a morphological error, they are there as well. If you want to find a histological error, they are there as well. But just to give you one example, that even the behavior as complicated as learning has also been affected.
Thank you, sir.
Aida, please go ahead.
Okay, so my next question is a bit off topic to मतलब ये कहना चाहिए कि अभी तक तो हम ड्रोसोफिला की बात कर रहे थे. ये टॉपिक मैंने फिक्शनल जो हमारा जो क्लब है, फिक्शन क्लब, हम बुक्स रीड करते हैं फिक्शन की. So from that area I'm coming. So question is, new biological civilization. There's a book on Amazon, a fictional book, Out of Control, Rise of Neobiological Civilization, and like that. So I googled it, but there's not much on the Google about it, no? And there's a Kevin Kelly, also there's an author of Kevin Kelly, Rise of Neobiological Civilization. So what is Neobiological Civilization, or is there any, like, is just a fiction, or is there something in books or journals as well about it?
So one thing I possibly may have not heard you clearly. It's a new biological civilization or neo-biological civilization?
NEO.
Neo-biological civilization. All right. So I never came across with this book. So all what I am saying is possibly you may say I am giving my opinion just reading the book by title. So that may not be, the book is not all about it. But what the term neobiological civilization means, that does make sense. And we are into it. And in fact, in every single generation, we are into it. So when we use the word neo, or when we use the term neo, neo means retaining the ancestral characteristics, tinkering it a little bit to make a new characteristic. So any offspring, they can take the characteristic from their ancestor, from their parents, if you like. And if they have taken the characteristics from their parents, they may run it as they like, as it is. Like whatever the parents are thinking or whatever the traits the parents have, they can take it forward as it is. Or they may tinker it as per their need or as per their convenience or as to be more aligned to whatever the society is dictating or whatever their needs are dictating to them. Right? If this be the condition, then they modify those traits to some scale. The offspring modify the ancestral trait to some scale. It could be a behavioral trait. It could be a morphological trait. It could be anything, any trait you like, any characteristics you like, which you take from the parents. And if you modify an ancestral trait to make it a new one, that's where we use the term neo, N-E-O. Okay. To me, every single generation is a neobiological civilization. Consider the way you are talking to me. Possibly you are on the laptop. I am talking to you on a laptop. Okay. We both are talking into a laptop. You may be 20, 30 kilometers right now from where I am being stationed. Okay. You are watching me. I am watching you. I am listening you. You are listening to me. Take up this entire theory or take up this entire experience of yours and go back in time about 250 years. You may find your great-grandparents. Okay. And you tell, this is what happened. And this is what not only happened and not being considered as a miracle, rather than a common thing. They would burn you for heresy, or they would believe that you are a demigod. These are the two results, all right? So that's mere 250 years we are talking here. And similarly, you tell your parents, you give your smartphone to your parents, and look how effective they are in using that phone. Initially, when you might have given them, and I beg your pardon, and with all due respect, it looks like a chimpanzee of a phone. They're simply unable to use it. And still, I do have a smartphone. I am being trained. I have worked on bioinformatics, and still I got few functions which I resolved few days ago by one of my BS final year student, because there was a live wallpaper which is coming again and again on Meghan Markle. And I don't know why it is coming. And my wife is a bit doubtful about it. So she was saying that why you are having Meghan Markle all the time on your phone. I say, I don't know where it is coming from. So then I don't know how to get rid of it. So I consulted to a person which is half an age than mine. And it was on her fingertips to resolve that problem. Don't you think so? That's a new biological civilization. I perfectly think so. So Yeah, every single generation is a new biological civilization. And you would find a difference between them as you go more distant. In fact, their degree, like still our parents hold some of the thoughts which our grandparents have. You hold much lesser thought than your grandparents have, or you consider some of those thoughts as being viable enough, but you rejected most of those thoughts. The next generation, your kids, are gonna see you and your grandparents and their grandparents as being completely a nutcase. This is what we see. Look, now you do not take, be a bit slow in realizing those things. But consider 20 years ago, LGBTQ, they are 20 years, just share 20 years ago, is being a full cardinal sin and a crime in many of the American states. They moved to the then they modify it and modify it and modify it, and now it's a word celebrating. They have a Pride Month or Pride Day. From a sin to being proud about it. It takes a journey of 20 years. So anthropologically, yes, we are into a new civilization, a new biological civilization, and we are all the time in the new biological civilization. And the rate of converting a civilization into a new biological civilization is exceedingly fast as we move along because of the technological advantage which we got. This is a smartphone. You take this smartphone and you go to 30 years when the peoples might have Nokia or some big phone, big cell phones, and show it to them. They would be amazed and looking at this. So we are in the new biological civilization, and we will always be. And the speed will increase, I believe so. But that's, again, reading the book by cover. I have not read that book. I never came across with that book. So I believe possibly that's what they are talking about.
Excellent. I recently read a book. It was Alvin Toffel's book. It was Future Shock. And it was published 52 years ago. Title is Future Shock. And it talks about the development in research versus our social development, which is very slow. And the changes that we are facing, just like you mentioned, one of the case, it is too fast for our slow brains to adapt to. So then you can have two reactions. One is we should be fast. And the other is no, that is sin. That is evil. I'm going to live in my own cocoon. So there are two reactions. And what you are suggesting is we need to adapt to different situations. Just like evolution-based adaptations led us to survive in the 21st century. And even before that. And we can now change our climate and other things, right? So excellent food for thought. Now we can, yeah, sure, go ahead.
There is a problem in it, and it's possibly the
Dr. Mushtaq, I'm getting some distortion in your voice.
Yes, it's a quote. Yeah. So there is a quote of Imam Ali, and possibly in a book called Najul Balagha. And it says, humans are fundamentally the enemy of knowledge. So that's the book I was talking about. Yeah, this is the book. So why? Because the knowledge actually make them realize how feeble and weak they are. any new knowledge. So let's take this concept at the first place. Second, most of the technological, now we know that the humans, as far as the heights are concerned, consider the heights of the human. So you would find there are very few short individuals. Take any population around the world. You'll find very few short pupils. Then the majority of the pupils have a medium sort of an height. And then very few are the extremely tall individual. So always there will be a normal distribution, a bell curve. Few very short, few very tall, and majority of the population is of the average height. So it's a bell curve, okay? Same, and those average pupil generally see those tall pupil and those short pupil as being freak, more or less. If considered they are not being educated enough. This is an African tribe, which is completely out of the civilization, modern day civilization, if you like. So what, I may not be very politically correct here. So, and pardon me for that. I don't want a Twitter account to, at the rate, saying this must be said. But this is what exactly is. Your height distribution is like that. Take any population. You will find majority of the peoples who got, if you go with the disease burden, that whatever kind of a disease, you will find very few pupils who are always be sick. And then you will find very few people which are never be sick. Okay. And then you find majority of the pupils who are being sick once in a while, in 1 year or 2, or maybe 3. Okay. That's for the sickness or any illness. That's for the height. You will find very few people who are extremely dark in complexion. You are finding very few people who are extremely fair in complexion. And then you will find an average wheatish color in the Pakistani population. Okay. So this is how our population is. The general population has a normal distribution. And generally that average population, which is generally in the majority, consider both of those extremes as being freaks. Some sort of freaks. Or freakish, or little odd, or weird. Let's say, more politically correct, weird. Okay. Now, same is a spectrum for the human intellect. There are very few extremely dumb people, and there is no polite way. But face it or not, there are few of us which are dumb. And there are very few which are extremely smart people. And majority of the people lies over here at the central part of average intellect. Okay. Usually the discoveries are made by individuals who are at this pole, which are extremely intelligent. So call it this guy, Tesla. Do you think so that person would be of average intellect? I don't think so. Call it this guy. You're already aware of it. Einstein, not of the average intellect, surely. Or maybe this guy. Isaac Newton, absolutely not. Or this guy. This lady, MadameCurie. They're not of average intellect people. So this progress or new knowledge is being created by those weird individuals. So one could expect that the central mass, which were even not very comfortable with an extremely tall or extremely short individuals, extremely dark or extremely fair individuals, how would they be comfortable with those weird pupils are providing them a new knowledge that made them realize how feeble they are. So the subconscious mind, in my view, the subconscious mind is going to challenge them. And there are very few civilizations who overcome. They are, now the rate is comparatively increasing, that's fine. But like Europe is very okay with it. They understand. There are very few individuals who are extremely sharp, they are scientists. Let's follow them, because the last 200 years is telling us that if you follow them, you are on the path of the progress. You can build drones, you can build satellites, you can run your cities without any shortage of the electricity, clean water, your disease burden is going to drop. They are the one who are going to cure my diseases. They are the one who are going to prevent me from any kind of the pandemic. Let's follow them. Most of the European countries have came across with that. So they have learned it and they are doing it fine. So their GDP and everything is a pretty much their disease burden, their society happiness is pretty much an indicator of those things. Compared to that, there are few civilizations, even in the developed countries, who had a problems. And again, it comes from the personal truth, social truth, and objective truth. Like in the America, we would find extremely large proportion of the population who are denying, large proportion of the population who is actually denying, in fact, the existence, the very existence of the virus SARS-CoV-2. There are a large proportion of the population who are simply denying the efficacy of the vaccine. It's much larger than in Pakistan. You would be surprised. In America, forget about Pakistan. Okay. They are seeing it, that their own kids dying out of it. They themselves are getting sick, but it's still not believing it. And it's still dying out of it. The highest mortality rate is there in America. And it's been considered the country where the most effective vaccine against the virus is actually being developed. That's Pfizer or Moderna. And that country's own population is not agreeing with it, because the vaccine has been developed by the individual, more or less, who are at this extreme, the weird individuals. And they don't like to be taught, isn't it? They said, oh, no, I know my stuff. And give some of those things, some of your opinion. You might have faced every time, at least once in a week, with your parents, exactly the same thing. You're telling them something and they say, oh no, you just don't know. हमने धूप में बाल सफेद नहीं करे, हमारे बाल ऐसे ही नहीं गिर गए. Where it is coming from, by the way? Well, I may not be very politically correct. So I'm going to switch off my Twitter account, which is not existing, or Facebook account for a month. So troll me as much as you like. But
no, that's absolutely fine. Yeah. As far as the extreme end of the spectrum is concerned, you can say in terms of IQ, let's say, you showed us beautiful models of Tesla, Einstein, Newton, and I can see the Darwin's model behind it. Yeah. Out of these 4, 5, 6, there was one female model, right, of MadameCurie that you just showed us. So I want you to comment on the gender parity in terms of intellect or research. Now we know that there is no gender parity in terms of intellect, but as far as research output is concerned, there used to be gender parity. And then you can talk about your own experience, whether your students, they are mostly male or female, and the prospect of research in Pakistan in terms of, gender parity, the differences between genders.
Now, again, I may not want to be a Jordan Peterson, but there are few things. let's face it. Let's face it. If somebody would say me that both are equal, they are demonstrably wrong. It's just simply not possible. The DNA is not equal. How on the earth you are saying the chromosomes are not equal? How on the earth you are saying they are equal? They can't be equal. They are not biologically equal. Let's not talk. Let's not do those things. And that's where the problem is. Our university were used to teach distasteful things. They are supposed to educate masses rather than being educated by masses. So that's the spine we are losing. Don't know. It's new age buzzwords which are coming into existence. And without thinking it through, that's it. We are adopting it as being a symbol of being very modern or very enlightened. This is not what enlightenment is. The enlightenment is that you have a freedom of thoughts, and then you have a freedom of opinion, and you go with wherever the evidence are leading you to. And the evidence are overwhelming. I'm not saying one is superior than others. That's what not I am saying. What I am saying is that I'm not saying one is superior than other, neither the female or the male. What I'm saying, they can't be equal. Simply, their biology is not equal. Their organs, in some cases, in very parts of the organs are not same. Their expression of the genes are not same. Check out the normal serotonin level in the humans, the male serotonin level and the female serotonin level. The chemical composition of the very neurotransmitter is slightly different between both of those things. Those neurotransmitter, Jamshed could tell much better than me, are defining your behavior. So they simply can't be equal. They are different. Not again saying one is superior than others. That's what not my point is. So they are different. Now, you could use them, or they could use their specific attributes where they are good at differently, where they could be best placed. So as far as gender inequality is concerned, yes, it was existing at one point of time. It was very much existing to certain sectors of the life at this moment of time. But as far as science is concerned, it's long gone now. It's been gone very much so. Now you may find occasional cases of gender biasness. But let me tell you, if you dig them very specifically, if you really dig them very specifically and unbiasedly, I'm sorry, being a harbor of a Y chromosome doesn't make me always wrong. Okay, and doesn't make me that token could be simply reversed because somebody is harboring 2X chromosome. They are always be right. How is it possible? So you would find, and there are cases after cases which are coming into the existence, which are telling us that if there is a gender inequality, 50% of the cases either being misconceptions or justifying one's own lack of talent or lack of progress by simply saying, oh, I have been discriminated. In this scientific world I am talking about. It was there, but not anymore. Not very much so. As far as my lab is concerned, now you can see here, there is an overwhelming, this is Tuba. This is Tuba here, Muqaddas, Hafsa, Aisha, you have already seen. And then there are two males, Tawseek and Zahid. And then we have Hansa and Fahad and Shoaib, Anusha there. Don't know what is a central girl, and there is an effort. Center mein kaun hai? Rabia is in the center. So there are quite many males, quite many females there. I'm sure there is still be a gender biasness in the professional life and elsewhere, but it is not of that scale that you should be too much worried about it. At this moment of time, if you are being outshiner, you have been shined back in year 1912. And if you are not an outshiner, then you can marshal all sorts of reasons. You may be, there are some injustice might have taken place with you, but there are similar kind of the injustice which have been taken place with your male colleagues as well. So do not pin every injustice you might be facing. It's just because you are being a female or being a male. It's not always be the case. You might be feel yourself being unjustifiably dejected, but it doesn't mean that it's just because you are being a female. And obviously, there are some gender inequality there. I agree, but it is not that scale to which we have made a hype of. And beside this, we have also made it something like of a status symbol, a symbol that if I am not talking about the female rights, or I am being actually slightly criticizing the feminist movement, or I am slightly criticizing all the arguments which they raise, they need to be believed in, then I must be old person and person from a middle ages or maybe bronze ages. It doesn't make me that. It just as much as they have a right of saying their opinion, let them be, because they consider themselves to be a liberal. And that's absolutely fine. They should cherish themselves. But liberal means they have to allow the other person to state their opinion, isn't it? And when you are going to state your opinion, you are going to be offensive to others. The scale of the offensiveness may be varying here and there. And if you are going to say this something and you disagree with some position of somebody, you're going to offend that individual, and let's have it. Let's tolerate it to be get offended if you are going to state such things. So that's my personal opinion as far as the gender inequality is concerned. In fact, there are some positions, and I was in one of the meetings a few weeks ago where Azra Palijo was there, who is a minister for the education, I think so, provincial minister of the education. And we were in that meeting, and she was saying that they are seriously thinking about establishing a male medical college now. Looking at the consequences, female. There's so many female medical colleges we have established just because, oh, your females are being we wasted a huge amount of money on that, and there is a need for some of the female medical college. That's fine, but the overwhelming numbers we have made, and now we realize that, oh, we made a mistake. Now we are short of male doctors, and we already saying 80% of the female doctors are not going to continue their education. And not going to be a physician. So it's a waste of all resources happening in our society. So you will find, I don't think so there is an injustice which has always been gender based, and in the field of the science, it's long gone now. It's long gone. It might be there, but not of that scale.
Some.
Hello, Dr. Mushtaq. Are you there?
Yes, yes.
Okay, we lost you for a few.
Yes,
For
yes,
a few moments.
I realized my internet was not behaving nice today. It's misbehaving a little bit.
Okay, okay. No problem. Should we continue?
Yeah, yeah. So,
Okay,
sorry,
so we can. Okay, okay. That's not a problem. We can continue with the topic that you were discussing. You were discussing gender parity, and you mentioned about Miss Azra Palijo, that there is the reverse trend can be seen in Pakistan even. So
Yes, yes.
yeah, so that's excellent that you were talking about. Sundus, you have some questions, so you can unmute yourself and you can ask the question.
Assalamu alaikum, sir. So my question is that research requires a good teamwork, right? And according to me, good teamwork makes dream work now. So sir, what is that one thing that a good team should have, और आपकी टीम में ऐसी कोई quality होना चाहिए, quality है, or is it hard to find passionate enough people for research?
It's hard to find, number one, the teamwork, what I believe is that if they share the same level of patient, both are the intertwined kind of a question. But Sundas, if you don't mind me, can I be excused for a minute? Because I just want to take care of my cat.
Yeah, sure. Go ahead. Go ahead. Go ahead.
Yeah, all right. I'll be back in a minute.
Meanwhile, students, you can write down your questions. You can write down in the chat section, or you can WhatsApp me. And then if your video is on, that's preferable. But if you don't want to keep your video on, that's also fine. You can ask your question directly to sir. So he can answer as much as he can. And the questions can range from evolution to research, to even some of the social issues that may be related to research and public understanding of science. So Dr. Mushtaq is very well versed in those subjects, and he's also very well read. So take maximum advantage of this opportunity. Okay, so I would allow Sundas to continue. And then afterwards, we have Areej from Salim Habib University, and then Rashid. They can ask questions. I don't think I need to elaborate more on the gender parity question. You can't hide behind your lack of talent by saying that there is gender parity and misogynistic culture. The patriarchal culture might very well be a true thing, right? But you cannot make this an excuse for not excelling in your field. You should at least give your best. So that's what I think Dr. Mushtaq, means to say. So you can agree or disagree with him, but that's what he's saying. And I partly agree with him that victimization leads to a defeatist mentality. And if you have that mentality, that no matter the resources and the opportunities you are given, you are always going to feel yourself like a victim. So if you always feel yourself like a victim, and in the West, he mentioned that it has become a status symbol sort of thing, that how much victim you are. I'm a victim, therefore I am. Not the initial Descartes thinking that I think, therefore I am. So it has become, I am a victim, therefore I am. So instead of dividing ourselves into binary thinking of victim versus oppressors, we need to try to muster our courage and excel wherever we have the opportunity to excel. And then you can think about, creating opportunities. And if you are given opportunity, please try to make room for others as well, right? So that's what my 2 cents on what Dr. Mushtaq mentioned are. And he did talk about, he just mentioned Dr. Jordan Peterson. So his thoughts, you can go online and type his name Jordan Peterson. There you can read about it. And then his one of the book, 12 Rules for Life. So it's just a general guideline how to spend your life. And then his another book, Beyond Order, which is much more philosophical. And before that, he had written one more book, which was Maps of Meaning. So Maps of Meaning is I would say, quite a dense book. It's not a good approach to listen to the Maps of Meaning. You should have the physical copy of it. Then you will be able to understand it. I think I have got Dr. Mushtaq back. Dr. Mushtaq, are you back?
Yes, I am.
Okay, so you were talking about Sundas's question. If you like, Sundas can repeat the question.
The team. No, I remember that question. Okay. What are the fundamental ingredients to be present to be an effective team player? If I want to rephrase the question. Well, one of the important things is that if they had holding same objective in the mind, okay, and there has to be a very effective leader to be present. So say, for example, if you might have seen herd of sheep, एक बहुत अच्छा जो चराने वाला होता है, I can't remember the exact word for that. Maybe Jamshed could have. Shepherd. Yeah. So if an effective shepherd can make all those sheep as a good team, they always move in a single direction or as the direction being told. There has to be one very effective leader. I would like to quote, और ये मैं अक्सर करता भी हूँ यहाँ पर, कि एक मूवी शायद आप लोगों ने देखी होगी, चक दे इंडिया. उसमें वो लड़कियों की हॉकी की टीम होती है और वो आपस में लड़कियां लड़ रही होती हैं. तो उसमें जो सबसे ज्यादा मिस्चीवियस लड़की होती है, तो शाहरुख खान एक दिन उसकी हॉकी पकड़ के कहता है कि और एक बात सुनो, हर टीम में एक ही गुंडा होता है और इस टीम का गुंडा मैं हूं. So you need one kind of a gunda, if you like, in a team, especially if the team is of Southeast Asia, because where the pupils are generally have a different approach what a team is supposed to be. बाकी आप क्लैप ट्रैप वाली टीम वाली बातें बहुत सारी कर सकते हैं. Oh, team has to be a coordinated one, team has to be this, and the leader should be that, and leader should be that. Give me one of that leader, actually, the name of one leader which you read in history, which is not tyrannical in at least some of his approach or her approach. Give me one. You may not find any single one. There are points where somebody has to be, the leader has to be assertive. The leader has to take the decisions which are generally disliked by majority of the team players, or majority of the components of the team player, or majority of the masses, or so for making an effective team, one of the things which is being extremely necessary is the leader who has a courage to take decisions which are not being well liked, which might be right, which might be wrong. And he has a courage to face the failures, and he is able to live with what we called the success. And he has an ability to, one can say, expand the credit of the success to the team workers. So these are the 2-3 things which are being extremely required, an effective leader. But if you are going to say that a leader has to be always be very polite, always be very encouraging, always be very thankful, well, I'm sure that I won't see a single leader in the history of the world who is like that. And if you had a name in your mind, or if you have read about any such leader, do let me know the name, educate me. I've never seen one. I've never read about one. They have to be polite, but there are phases where they have to be very, very assertive. They have to be tyrannical, or they have to be dictator. So that's one. The second thing is that the team player, the other components of the team, they have a single passion or objective in their mind, and they are a bit more selfless. And obviously you will find individuals amongst them who are a bit selfish, who are not in the same frequency. Then you can always mold them. Okay, look, you can always make them realize the success depends if all police are working in the harmony. And if you work in harmony and in a synchronous way, you may travel the distance, which is at this moment of time, looks beyond imagination, looks very difficult. But it all depends if all police are working in synchronous way, in harmony. If one of them get rusted, you can get it corrected. The leader has a job, or the other police have a job to remove the rust from one of the police. And if you see a pulley is actually deteriorating, a wheel or a pulley or any component or any gadget which has been present in the machine of a team being rusted enough or being not effective enough, and there is a chance that it can damage or it can deteriorate the overall productivity of the team, the leader has a courage to remove or replace that part for good. So this will make a good team. And if you ask me finding a good team, let's say I started a very big experiment, and at the very big experiment, I said to, I made a team of about 12 or 16 individuals. 12 से 16 बंदे थे उस टीम के अंदर, ठीक है। और इसमें मसला ये था कि हमें 24 hours लैब में रहना था, आप ये समझ लो, for about good 2 months, 2 to 2.5 and 3 months. It's a project of 2.5 to 3 months. We have to be in the lab 24 hours. Obviously, कोई भी बंदा 24 घंटे लैब में नहीं रह सकता, ठीक है ना। सो फिर उसको हमने टीम में डिवाइड किया हुआ था कि कुछ पार्ट ये कवर करेंगे, कुछ पार्ट ये कवर करेंगे, कुछ पार्ट ये कवर करेंगे। और जो रात वाला पार्ट था, जो कि स्टार्ट होता था 12 बजे से लेके रात के 3 बजे और फिर 2 बजे और फिर 2:30-4 बजे, वो वाला पार्ट मैंने अपने जिम्मे और मेरे 2-3 स्टूडेंट्स थे उनके जिम्मे दिया हुआ, ठीक है ना। We were working it all fine. We started with 16 individuals. We left with 8. So you will find individuals dropping by here and there, and that is very normal. And at that moment of time, the team or the leader must have an stamina or mental stubbornness or mental strength that if that instrument is missing, if that part of the team is missing, I am not going to collapse, or I'm not going to just change the plan. I'm going to continue with the plan. May my speed get affected, but I will transform this load to the other components of the team and myself as well. And that makes a good, effective team to be very productive. So yeah, you may find individuals. There are some crazy individuals over there. I got lucky. I got one individual over here. She, her name is Ifat. That's Ifat. Ifat. That's Ifat. You know, so Ifat was a fresh pass out of BS Biotechnology. Along with her, two of the very smart girl, very hardworking girl, Aliza and Munaim were there. They were the thesis student. Ifat actually lacks 1 or 2 marks, but she volunteered to work in the lab. She worked effectively well. And what transformed, in my opinion, transformed what I witnessed from the work ethics and the working ability of the Ifat. I would say that if I have to rank out of the 40, 50 students which I got of postgraduate of all time, if I have to rank the top 5, Ifat is surely one of it. So again, the leader or the individuals within the team has an ability, and that's not my only opinion. That's the opinion of all the major component of the team which I made for that small project. So the team members should have an ability to realize the potential of one of the component. Oh, we place that component there, but if we place that component there, he or she would be mighty effective. And we all realize it. So that's another thing. So 3 major things: effective leader. The leader must have an ability to make unpopular, distasteful, often to some scale dictator type or tyrannical type decisions. Number 2, the members should be receptive enough and working for a common goal and have a stamina or aptitude to tolerate or failures. And you always have a backup plan.
Excellent. We have Arij with us from Salim Habib University. Arij, you have a question. Please unmute yourself and you can ask sir directly.
Hi. First of all, Professor, it has been an honor talking to you. And the session has been really, really informative. And I'm very thankful to Sir Jamshed for arranging the meeting. Sir, I'm curious about a question that has your Drosophila research resulted in any understanding of neurological disorders and muscular dystrophies in humans, especially in children with cerebral palsy? And also, what makes you so different or special researchers than others?
What makes me very different from the, the second component is very easy.
Okay.
You said what makes my research be different than the other researches which have been conducted on Drosophila? That's what your research, that's what
No,
you're saying?
no, I'm saying what makes you a better researcher or a special researcher than others?
Well, what makes me a special researcher? Possibly I'm the worst one. That makes me special, if you like. So I would not categorize I am being special enough than any other of my colleagues. To be very honest, I personally think so that I am sort of a mediocre kind of a researcher.
Look, sorry to interrupt, but actually, Sir Jamshed, mentioned that you have been selected in some university in, I guess, US. So I was very curious about that.
Yeah, I am a visiting professor at the Loyola University at this moment of time, at their health science division. So I am at this moment of time, I have to spend about 3 months a year in Chicago, since having a professor over there. And what I'm leading to is some of the researches which are conducting, which have a component of bioinformatics, as well as the components about SARS-CoV-2. So that is what I'm supervising, about 3 PhD students there and 2 MPhil students there at this moment of time. I'm a part of the supervisory team. So that's one thing, but that doesn't make me special. It could be achieved by any other individual. So I have nothing special except one thing which I am being proud of. I'm not saying that this is an exclusive quality which I got. I want to work, if you like to put it in this way, with the extreme degree of self-reliance, if you like. So one of the things which I do, or I did actually, that's not, my university is not going to support me. My university is fully willing to support me altogether. But one of the things which I put that barrier on myself, that I will make my own lab out of my own money, whether I take that money from the grants, whether I take it from my bank account, whether I get it from any other kind of the businesses which I am running. So whatever the labs which I got, they are mine. Each instrument which is coming in that lab, or machines or consumables which are coming in the lab, they are all mine. I'm not taking anything from the university. And that allow me a bit of a freedom to work whatever I like. So I can risk quite much of the things. And that makes me more, more personally connected with any equipment, any consumable, any work which I am doing in my lab. So it feels home when I am here, because in your home, all the things are possessed or being owned by you. So if in your lab, all the things are owned by you, then you will feel that the lab is your home, isn't it? So that possibly make me slightly special, if you like, but may I not be very exclusive in it. That's one. As far as intellect is concerned, I am averagely smart individual, and I still learning quite a lot from, in fact, learning most of the things from my students. So they are the one who are actually guiding me through what to read and how to think in many cases. In some cases, maybe it's other way around, but in most cases, the information is imparted to me rather than I am imparting the information to them. Or that's nothing special about me. The other thing which you asked about the other researches which with reference to the myotonic dystrophy, or you say neuromuscular dystrophy, and cerebral palsy. Not sure about the cerebral palsy at this moment of time, but there are few hints which I have seen in some of our essays in the flies, which may indicate a possibility of neuromuscular disorders. I would not say at this moment of time dystrophy, because for that purpose, I have to run histological investigation, and which we have already planned it. So if you see, that's where Anusha is working. And right, Anusha, you may see right above Anusha, you may see there are boxes and boxes and boxes. these are the flies which we have stored out of our experiment, and these flies will be subjected to the stereo microscopy and then histological investigation of muscles and parts of the brains as well. So only in that, once that being investigated, then I could say very conclusively, there are some neuromuscular disorders, but at this moment of time, claiming it to be either dystrophy or palsy would be a long shot. It's a far-fetched idea. But once those histological sections are in front of me, I may have a much resolved answer for your query.
Okay, good. We have taken a lot of precious time of Dr. Mushtaq, so I can allow only one last question. Please, Rashid, go ahead with your question. This is going to be the last question, and then we will bid Dr. Mushtaq a goodbye.
सर मेरा लास्ट क्वेश्चन ये है कि आप ड्रोसोफिला पे रिसर्च कर रहे हैं और आपकी जो फाइंडिंग्स आ रही हैं, जो आपके रिजल्ट्स आ रहे हैं ड्रोसोफिला से रिलेटेड, आप जो जो एक्सपेरिमेंट कर रहे हैं, आप जो जो चीज़ें ऑब्जर्व कर रहे हैं, एग्रेशन आपने बोला कि आप देख रहे हैं और भी काफ़ी पैरामीटर्स आप देख रहे हैं, तो मेरा क्वेश्चन ये था कि अगर हम कंपैरेटिव स्टडी पे जाएं तो एक ऐसी, ये तो आपने एक लैब में सेटअप बनाया हुआ है ना, पूरा आपने प्रॉपर इन्वायरमेंट दिया हुआ है, न्यूट्रिएंट एगर सब कुछ, एक अगर हम प्रॉपर इन्वायरमेंट आउटसाइड हम जाएं, लैब के आउटसाइड, वहां पे जो डेजर्ट में ड्रोसोफिला मौजूद है और बाकी जगह पे यानी कि वॉइड में जहाँ पे मौजूद है, वो एक नेचुरल इन्वायरमेंट है जहाँ पे वो मेटिंग कर रहे हैं और बाकी ये सारा, तो उसमें जो जेनेटिक डाइवर्सिटी है और आपकी लैब के अंदर जो फाइंडिंग्स आ रही हैं उसमें जो जेनेटिक डाइवर्सिटी आ रही है, तो उसमें आप क्या कमेंट करेंगे?
Oh, wow. Quite clearly, that's been true for any study, if you like to do. Say, for example, if you say in a population, in an American population, a particular allele or a particular mutation, a particular variant of a gene is being present at that percentage, let's say 0.2%. So it means if you take 500 individuals of that population, you will find one individual who have a mutation, right? 0.2% allele frequency represent that. But having said that, what you say, should that research have actually screened the entire American population? The answer would be simply no. They have taken a representative sample. Any study you do on mice, should that represent the whole population of mice around the planet Earth? No. That's under the laboratory condition, because the laboratory condition are supposed to be the standard condition. So as far as the genetic diversity is concerned, obviously in the natural habitat or on this very blue dot, you would find much extensive genetic diversity of the Drosophila compared to what's being present in the lab. That's absolutely fine. But think about it, that makes us more advantageous in a way. Why? Because let's say हम एक experiment कर रहे हैं inbreeding का, जिसमें मुख्तलिफ तरह के cousin crosses और relatives आपस में, fly के relatives आपस में cross कर रहे हैं. जिन families के अंदर cousins में ही marriages होती है, उनमें ये पहली नस्ल नहीं होगी जिनमें cousins में marriages हो रही है. उनमें एक अरसे से cousin marriages चल रही है. Look around it and you will find कि उनके दादा भी आपस में कजन थे और उनकी नानियां भी आपस में, नानी और दादी भी, वो भी कजन थे और ये भी कजन थे और वो भी कजन थे. And then you will find a population, एक इतनी बड़ी population से एक छोटा सा section निकला है जो कि आपस में cousins marriages करता आ रहा है. ठीक है. Now that actually simulate in a way a real-time environment where we would find inbreeding is a problem. Say, for example, in America, if you go with Amish, Jamshed, correct me if I'm being wrong.
Amish culture. Amish culture.
Amish culture है या Amish community है. अच्छा, वो आपस में ही inbreed करता है. अब ऑब्वियसली अगर आप बिल्कुल बैक ट्रैक करते जाएंगे तो ये आमिश कम्युनिटी जो होगी ये किसी वक्त में एक वाइल्ड टाइप, एक बड़ी पॉपुलेशन से सेपरेट हुई है, और सेपरेट होने के बाद वो आपस में क्या कर रही है, इनब्रीडिंग कर रही है। सो आप ये नहीं कह सकते कि आमिश कम्युनिटी में आने वाली रिसर्चेस या आमिश कम्युनिटी में होने वाली ये जो कजन मैरिजेस या सिबलिंग मैरिजेस या रिलेटेड क्रॉसेस की जो रिजल्ट्स है वो पूरी अमेरिकन पॉपुलेशन को रिप्रेजेंट कर रहे हैं, नहीं, लेकिन वो आमिश कम्युनिटी को जरूर रिप्रेजेंट कर रहा है। ठीक है ना, चाहे आपने पूरी आमिश पॉपुलेशन को एनालाइज करा भी हो या ना करा हो, लेकिन उसके चंद एग्जाम्पल वो उसको पूरे को रिप्रेजेंट कर रहा है। तो इसी तरह पाकिस्तान में भी जिन खानदानों में कजन मैरिजेस हो रही है उनमें कजन मैरिजेस पहली नस्ल में नहीं हो रही होगी। अगर आप उनकी पीडिग्री ट्रेस करेंगे तो आपको पता चलेगा कि वो बहुत पहले से आपस में कजन मैरिजेस करे जा रहे हैं। सो अब अपने वाइल्ड टाइप पॉपुलेशन से एक सब सेक्शन ड्रोसोफिला का निकाला और उसको कजन मैरिजेस की तरफ ले गए। सो इट वुड बी अ रादर बेटर रिप्रेजेंटेटिव टू व्हाट इज हैपनिंग इन द ह्यूमंस rather than just go in the wild type population and start screening those flies. Okay? अच्छा, उसमें भी हमने एक चीज का बड़ा ख्याल रखा है कि उसमें एक कंट्रोल भी चलाया है। वो कंट्रोल ऐसे ही चलाया है कि जिसमें जो फ्लाइज आपस में क्रॉस कर रही हैं वो आपस में रिश्तेदार नहीं है। एंड हाउ वी मैनेज इट वो होपफुली जब पेपर आएगा तो आप लोग पढ़िएगा, एंड देन वी मे हैव अ नेक्स्ट सेशन where we'll talk about it once we have published the paper, कि how we make sure कि ये आपस में रिश्तेदार ना हो, how we make sure कि ये आपस में सिर्फ बहन भाई हों, how we make sure कि ये आपस में cousins ही हों, how we make sure कि ये siblings में होने के बाद रिश्तेदारों के बाहर शादी करके फिर दोबारा siblings के साथ क्रॉस कर रहे हैं, how we make sure कि ये पहली दफा sibling बने या continuously sibling बने, and so on and so forth.
Thank you very much, sir.
Thank you, Dr. Mushtaq. And it's very good news that you are willing to give us your time, even in the future. Zabardast. On behalf of our fiction book club, thank you so much. And now I am going to click on the end meeting for all. So meeting will end abruptly, so don't be surprised. Thank you,
Okay.
Dr.